Hi, welcome to today's class. For today's learning purpose, we're talking about bacterial inoculation. Um, I want to thank all those that have been subscribing so far to our page. And if you are not yet subscribed to our page, I want to encourage you to please subscribe to our YouTube channel, like and share these educative videos for others to also learn from. So for today's learning purpose, we're talking about bacterial inoculation. Earlier on, we spoke about how to prepare culture media. Now, I want to talk further on how to actually culture on this prepared culture media. Now, how to inoculate the samples or organism into this culture media. So, there are several ways in which you can actually inoculate or culture microorganisms. The first is known as standard trick. Uh, the standard trick method. There are several ways. We'll be dwelling on standard trick for today. Also, we have pour plate. You can use the pour plate method. And you can also use the spread plate method. So these are just some of the methods that we can use to inoculate our petri cultural media with our inoculum. So for today's learning purpose, we're focusing on standard streaking method. Why standard streaking method? Now I'll tell you the advantages or why you should do standard streak. Now you know that to identify microorganisms, you need to see these organisms in colonies. If you watch my earlier videos on bacterial identification, you find that to identify microorganisms, that's to do colonial morphology, you need to identify them in colonies. It's best to identify them in colonies. So now, how do we make these colonies? How do we grow these organisms to be in discrete colonies? Because if you don't do standard streaking method, you won't get these organisms in discrete colonies. That is, they would not be one. In layman's term, they will all grow together and you will not be able to distinguish one colony from another. So standard streaking method helps you to obtain discrete colony when culturing, when doing your inoculation. So now, what are the steps involved in standard streak method? First of all, you get your prepared sterile medium in the petri dish. You know, from my earlier video, I showed you how to prepare what we have here is blood agar. I showed you how to prepare blood agar and then how to label and stack them in the refrigerator. Now, you bring this plate out from the refrigerator and you know that it's going to have moisture because you refrigerated at a temperature of 4 degrees Celsius. So now you know that this plate is going to have droplets because if you look at it closely, you find out that there are water droplets now on this plate. There are water droplets. So if you culture this inoculum now on this plate with water droplets, it's going to cause contaminants in the plate, and we don't want that. We don't want contaminants. So the first thing you do is to dry your plate using the hot air oven. Now the hot air oven uses dry heat sterilization to dry up the water droplets from this plate before you can now go ahead with your streaking. So the first thing we're going to do now is to dry our petri dish. Once it's dried, you will not see these water droplets around the edge of the plate and on the surface of the media. It's no longer there. Then you know you have dried. And drying is done at 65 degrees Celsius using the hot air oven. So now, you see, media is all dry, you see that there's no more droplets. There's no more water droplets around the plate and on the surface of the media. There's no more water droplets. So these are you dry your media. Now the media surface is all dried. It's ready for inoculation. So the first thing you want to do, this is your wire loop. So what we'll do, we'll sterilize our wire loop using our street lamp that's already on. So you flame the wire loop. You flame the wire loop red hot. Let me show you what I mean by red hot. So you have to flame till you see the down is becoming red hot. You see, it's now red hot. So the wire loop is red hot. You allow it to cool. We're not going to do this now. I'm going to show you on the board first of all before we practicalize it on the Petri dish. So let's assume this is our Petri dish containing the culture media now. So the first thing you want to do after sterilizing the wire loop, as I showed you, you allow the wire loop to cool, and then you obtain the inoculum. We are going to be using our urine sample for inoculum. So you take a loop full of the urine sample, for instance, as in you're using urine. You make a well of the inoculum. Now, when you are inoculating, avoid the edge of the plate. Avoid the edge. Avoid culturing on the edge of the plate. So now you make your well of inoculum. After making the well of inoculum, you flame the wire loop again. After you flame, you allow it to cool. Once the wire loop is cool, you streak from the well of inoculum on the Petri dish. You are not going back to the urine sample. You are going to streak from the well of inoculum already on the Petri dish. So I'll get my wire loop and I'll streak from the well of the inoculum. After you streaked, you flame the wire loop again. Then you allow it to cool. Once it's cool, you streak from the distal end of the previous streak. You are not going back to the urine sample. I repeat. You are going to streak again from the distal end of the previous streak. The distal means the ending part of the previous streak. So you streak from the distal end of the previous streak. This is it. Then you flame again. Once you have flamed your wire loop again, allow it to cool. You streak again from the distal end of the previous streak. The aim of flaming, cooling, and then streaking from the distal end of the previous streak is to reduce the bacterial population, because if you observe very well, the population of microorganisms that will be here will be more than the ones here. Will be more than the ones here. So the more you streak the distal end and flame, you cool, you are reducing the population of bacteria that you are dragging along the streaking line. So again, after you make this streak, you flame the wire loop, you flame the wire loop red hot, allow to cool, and then you streak again the distal end of the previous streak. Once you have done this is A, this is streak B, streak C, streak D. Once you get to streak D, you flame the wire loop red hot and you cool again. Note, you have to cool the wire loop. You must allow the wire loop to cool. If not, you will kill the microorganisms with heat. Maybe you are too much in a hurry, or you're not patient enough to allow the wire loop to cool, and then you went to streak immediately. You will kill the microorganisms if present. So that's why you allow to cool after flaming red hot. So once you are allowed to cool, you streak this time around in a zig-zag manner. And this is streak E. So if this organism is allowed to grow after 24 hours, you find out that the more get discrete colonies. So this is how we perform standard streak. It helps us also in identifying microorganisms, in picking out one colony to check for the cleaner morphology, the size, the shape, and all whatnot. It helps. So this is the essence of doing standard streak. So that is exactly what we're going to do right now. So now we have to flame our wire loop red hot. So I'll show you how the standard streak looks on our culture media. So now we flame the wire loop. Standardly, when you're culturing, you don't talk, so you don't culture your saliva. But for learning purpose, I'm going to be speaking so you can understand what I'm doing. So now wire loop is red hot. You can see the wire loop is red hot. So you cool. This is how you cool. You cool the wire loop. I know you may be wondering, how will you know when your wire loop is cool? You know, sometimes you can just touch the wire loop without any inoculum on the petri dish. You know, the petri dish is molten agar. So if it melts the molten agar, you know that it's still hot. So what I mean, I just touch it. If it melts the agar, you know it's still hot. So you just cool for a while. So once it's cool, you mix your sample properly, because sometimes if there are bacteria, if there are microorganisms, it may have gone to settle down. So you want to mix your urine very well, and then with these two last fingers, you open the lid of the universal container, and you don't drop it so you don't contaminate the sample. While holding like this, you dip to get a loop full. So this is a loop full of this urine sample. Then you cork. Then you make a well of the inoculum, just like I told you. So this is a well of the inoculum. Then we flame red hot again. Then we'll cool. We'll cool again. All we're trying to do now is to reduce the bacterial population. So we'll cool. So once our wire loop is cool, like I said, you streak from the well of the inoculum, not from the urine sample anymore. So we'll go to the well of the inoculum now and streak. So this is the well of the inoculum, so we'll streak. Now we'll flame again. Okay, so now you flame red hot again after making the first streak from the well. You flame red hot again. Then you cool. You cool. Then once it's cool, you streak again from the, like I said, from the distal end of the previous streak. The distal end, that's the ending part of the previous streak. So now that's what we're going to do. This is the distal end of the previous streak. So this is our first streak. So we're going to streak from the distal end. You see? You flame red hot again. You cool. Now once it's cool, you streak again from the distal end of the previous streak. So you see, this is A, B, C. We'll flame red hot again and cool. You see, you keep flaming and cooling just so you kill the microorganisms that got stuck to the wire loop from the previous trick, just so you could reduce the bacterial population and obtain discrete colonies. So we'll streak again from the distal end of the previous trick. See it? So you see, so we've streaked. So you see, this is the well. This is streak A, B, C, and D. You see how it looks. This is how it's supposed to look. Huh? So flame red dot again, and then we cool. Be patient enough to allow the wire loop to cool. Allow the wire loop to cool so you don't kill the microorganisms. That's why we wave our hands like this to allow air to cool it quite faster. Now we streak for streak E. We'll take it straight, distal end of D, and then we streak zigzag. So this is streak E. So at the end of the day, this is how it looks like. So you can see this is how it looks like. So this is how your standard streak should look like. Now flame a wire loop and then okay. So I want to believe you've learned something from today's class, and if you've watched this video to this point, why not subscribe for more educative videos as such. See you in our next class. Thank you.